Okay, so before this, we have seen how these chlorophyll are organized into what we call the reaction center and also light harvesting center. Okay, so long story short, okay, these are reaction center and light harvesting center. Ultimately, the function is to just collect or harness the energy from the sunlight and then use it for subsequent steps. Okay, so now we'll go to the subsequent steps, which are all biochemical reaction actually. Okay, so let's start from here. Okay, so this is okay, so maybe here. Okay, so for the whole process of photosynthesis, it can actually be separated into two stages. Okay, so first is what we call the light reaction. As the term implies, it requires the presence of sunlight for it to occur. And there's a second stage, which is a dark reaction, okay, which that can occur in the absence of light. Okay, so that's why the dark reaction is also called a light independent reaction. Okay, and of course, we see that this first light reaction is also called photophosphorylation. Okay, we'll talk about that in a minute. Okay, but long story short, in this process, later we'll see ATP is being produced. Okay, and actually the energy used to produce ATP is a light energy. Okay, so that's why it says light energy is transferred to ATP. Okay, so as we mentioned just now, your grana is where all the chlorophyll are, okay, and they function to collect energy from the sunlight, okay. So this case, okay, this reaction is called photophosphorylation, okay. So because, okay, when you convert your ADP to PI, okay, so you attach a PI to ADP, you need some energy, and at the same time, you kind of put energy, you stuff energy into it. Okay, if you remember in the case of oxidative phosphorylation, okay, we have this particular image, okay, so maybe I'll draw this again, okay. So your hydrogen ion is going to okay. So here, if you remember, outside the plasma, at the intermembrane space of the mitochondria, there is high concentration of H plus that kind of store electrical potential energy. Okay, so as hydrogen ion diffuse into the mitochondria, okay, so the electrochemical energy, okay, or the electrical potential energy is converted into chemical energy that is stored in ATP. Okay, so here, basically what happened here is that your electrochemical potential energy is converted into chemical energy. Okay, so that's what happened in the synthesis of ATP in the case of oxidative phosphorylation. Okay, and whereas in the case of plant, it's photophosphorylation because somehow this process, okay, the energy that is required for this process, okay, is provided by the sunlight. Okay, so you have sunlight, the light energy coming in, okay, and so you're converting light energy to chemical energy. So that's the process of photophosphorylation, which we'll see in detail in a minute. Okay, and the photoactivation of chlorophyll. Okay, so one thing you need to know, I'm not sure if you have learned that in physics already about photoelectric effect. So when something, okay, like a metal, for example, if it's struck by a light, okay, so it is possible that the energy absorbed by the electron from the light would cause an electron to be excited and then to leave the metal surface. Okay, so basically what happened in the case of chlorophyll is that when a light energy is absorbed, that energy is being is used to split water. Okay, so the energy that is absorbed by chlorophyll, the light energy absorbed by chlorophyll, is used to split water into hydrogen ion, oxygen, and electron. Okay, so this electron will absorb those energy, okay, and then it is going to move. Okay, so later we'll see how you will see there is some resemblance between the photosynthetic process and respiration process. Okay, so anyway, so it will give hydrogen ion and electron. So this NADP is going to, it will combine with the H plus and the electron, okay, to make reduced NADP. Okay, it is going to, okay, so again, so you have some hydrogen carrier just like your NAD in the case of photosynthesis in plant. This is NADP, okay, so as water is broken down by absorbing light energy from the chlorophyll, okay, water is split into hydrogen, ion, oxygen, electron. Of course, oxygen will be released immediately. okay, to the environment, so as a side product. Okay, so that's what we, at least from the perspective of other animals, this is what we want from the plant. Okay, and then the H plus and electron will be recombined and then accepted by NADP to form a reduced NADP. Okay, so the role of a light dependent reaction really is to produce ATP and reduce NADP. Okay, so later we'll see this. Okay, so let's see. So photophosphorylation is actually quite not to say complicated. Okay, so let's see. So there's a light pass to a reaction center. Okay, so as we learned just now, you have accessory pigments and the chlorophyll A. So all light energy will come to this reaction center. Okay, and a pair of electron is actually being is activated. Okay, so like I say, when energy struck, electrons will be excited from chlorophyll. Okay, and then the electron will be taken up by electron carrier. So again, okay, so electron that's excited, okay, it has high energy. It is going to move through a series of electron carrier. Okay, and then we'll see, okay, and then this chlorophyll molecule is going to lose electrons, so we say that it is oxidized. Okay, at the same time, okay, at the same time, just 1 minute, I want to see one thing. Okay, at the same time, NADP will be reduced. Okay, so actually later we are going to see this whole process is coupled with photolysis. Okay, so this note is a bit not very straightforward. Okay, but it gives you some overview. Okay, later we'll see it in detail again. Okay, but what you need to know is that at this level, okay, at the reaction center, energy is absorbed by chlorophyll. So electron of the chlorophyll is excited, okay, and then that electron will be transferred to a series of electron acceptor, okay, or on the electron transfer, something like an electron transport chain actually. Okay, so and at the same time there will be H plus produced, okay, again by, okay, there will be H plus and electron combined to form a reduced NADP. Okay, so later we'll see, of course, this part will be coupled with photolysis of water, because if you remember just now, water come from, the H plus come from water. Okay, so and then the electrons are going to pass along a number of electron carriers, okay, and then ultimately used to make ATP. So again, actually what really happened here, so if you remember, okay, so let's talk about this 1 minute. Okay, again, let's refer back to the process of oxidative phosphorylation. Okay, so let's a bit of a recap here. So this is a plasma membrane. So there are some electron transport, okay, electron transport chain. Okay, so I'll just call this an electron carrier. Okay, what happened is that here you have, okay, so if you remember the NADH, okay, is going to release the H, which split into H plus and electron. Okay, electron will be transferred across. Okay, so electron will go from here to here. And this electron has a so this electron carry a different energy level. Okay, so and then energy will be released. So in the case of a mitochondria, this energy is used to pump hydrogen ion. Okay, it's used to pump hydrogen ion. Okay, it's used to pump hydrogen ion into it's used to pump hydrogen ion into the intermembrane space, and then ultimately, okay, that hydrogen ion is going to diffuse down, and then you make ATP from ADP. Okay, so that is a process of oxidative phosphorylation. Okay, where you can see that the electron transfer ultimately is coupled with the formation of ATP. Okay, so here what happened in the case of in this case, okay, so what happened is you have the chlorophyll, okay, release electron, and of course it becomes I'll just use this chlorophyll plus, which means it has lost electron. Okay, so this electron is also excuse me. Okay, this electron is also transferred. Sorry. Okay, okay, also this electron is also going to be transferred, okay, used to pump hydrogen ion. And again, very similarly, ATP is produced. Okay, so that's why you see, if you look at here, so There is also electron transport chain in a thylakoid membrane. Okay, so the electrons coming from chlorophyll is going to be transported across it. Okay, and the energy release is also used to pump hydrogen ion. Okay, from stroma into thylakoid space. And that also again creates a steep hydrogen gradient or hydrogen ion gradient or proton gradient. So as proton diffuse into, diffuse down the electron chemical gradient, the energy is also used to generate ATP from ADP plus PI. Okay, but the only difference is that ultimately the source of energy comes from light. Okay, because this process require sunlight. Okay, there is light that provide this energy in the electron. Okay, in the case of oxidative phosphorylation, the energy, okay, so it comes from the NADH. That energy store come from a chemical source, okay, come from a glucose, so it's slightly different. Okay, so anyway, so that's photophosphorylation. Okay, so and then the electrons can enter either a cyclic or non-cyclic photophosphorylation. We'll come to see in a minute. Okay, but I want you to know, see the resemblance, okay, between, I mean, some of the similar strategies used to generate ATP, whether it is in mitochondria or in chloroplast. Okay, so but of course the difference here is that in mitochondria, I want you to know that hydrogen ion is pumped from the matrix into the intermembrane space. Okay, it's kind of in an outward direction. Okay, it's kind of pumping the you're pumping the hydrogen ion out from the matrix into the intermembrane space. Whereas in the case of chloroplast, okay, so it's actually pumping hydrogen ion from the stroma into the granum or the thylakoid membrane. Okay, and then as it diffuses down, diffuses back out of the thylakoid into the stroma, then the energy release is used to create ATP. But I just want you to see anatomically, okay, so the configuration are different within, okay, for the case of chloroplast and for the case of mitochondria. Okay, so now we see this photophosphorylation can again be separated into two kinds, okay, the cyclic and the non-cyclic. Okay, so the non-cyclic use both photosystem 1 and photosystem 2. If you remember just now, okay, so your chlorophyll or your color pigments are photosynthetic pigments are organized into photosystem. Okay, so one that's on the thylakoid, okay, or on the thylakoid membrane, and the other one or actually on the grana. Some are intergranule. Okay, so the intergranule are, let me see, where's intergranule? I drew it just now for you. Shit. Okay, so here, your photosystem 1 is here, photosystem 2. Okay, so photosystem 2 is here. Oh, sorry, photosystem 1 is here, photosystem 2 is here. Okay, so, but like I mentioned, it's not something very important. I mean, not the most important thing that you need to remember. Okay, so anyway, so what happened here is this, okay? So first of all, light energy, okay, so is actually, okay, so there's both your photosystem 1 and photosystem 2, they can absorb light from the sunlight, for example. Okay, so, and of course, when energy is absorbed from the sunlight, electrons are excited from the chlorophyll. Okay, so these electrons are captured by electron acceptor, okay, pass along the chain, okay, and of course, hydrogen ion, okay, energy released as the electron travel from the one electron carrier to the next at a lower energy level, energy is released, and that energy release is used to pump hydrogen ion into the thylakoid space, okay, and then so here you see the electron displaced from PS1 is replaced by electron from PS2. Okay, so now let's see. Okay, so here, so let's look at here first. Okay, so here your PS1, okay, so PS1 it gets excited, energy is, okay, electron is excited, or maybe hold on. Okay, I think maybe I'll start from here. Okay, I think we'll look at the text later. So if you look at this here, you have two systems here, okay, photosystem 1 and photosystem 2. Okay, light energy comes in, okay, like I mentioned, excite the electron. So the electron will be, okay, released, and then of course it will transfer, okay, it will be transported through the ETC, and then energy is used to pump hydrogen ion, and then hydrogen ion is used to generate ATP. That's something we already discussed. Okay, so same thing would happen to the PS1. Okay, so electron also get, also get excited, get transfer, and then pump hydrogen ion and then generate ATP. Same thing there. Okay, so the only thing, kids, so this particular point, okay, so if you remember, in the case of oxidative phosphorylation, for example, ultimately there will be an electron acceptor, okay, which will be oxygen in the case of oxidative phosphorylation. Okay, so here at least the electron coming from the PS2 will ultimately be received by PS1. Okay, so in a way you can say that the electron that is lost from PS1 will be replaced by electron from PS2. So that's why this particular sentence, okay, electron displaced from ES1, okay, is replaced by ES2. Okay, because this electron needs to be replaced, because if this chlorophyll is continue to work, whatever electron that has been lost, we have to come back so that it can be re-excited, and then again that energy can be used to pump hydrogen ion and generate ATP. So definitely electron need to be actually replaced. Okay, so in the case of PS1, okay, that electron come from that particular electron come from PS2. Okay, and then at the same time, some of the energy that is absorbed, okay, is also used to, okay, so some of the energy absorbed is also used to split water into hydrogen ion and electron. Okay, so here it would actually show, okay, water will be split into hydrogen atom and oxygen, and of course it will split into H plus and electron. Okay, so this is the H plus that is being pumped, okay, or diffuse down concentration gradient later to form ATP, and this electron will be accepted by PS2. Okay, so if you look at here, so what happened is that, okay, so electron lost from PS1 due to excitation will be replaced by electron lost from PS2. And electron lost from PS2 will be replaced by electron coming from photolysis of water. Okay, and this whole process, okay, so we call this a non-cyclic photophosphorylation. Later, you will see why is this called non-cyclic, because we'll see in the case of cyclic, it will be slightly different. Okay, so but anyway, so yeah, basically this is the whole process that we discussed about. Okay, so photolysis of water also, yeah, H plus, so the accumulation of H plus when they diffuse out of thallicoid space via the stop particles, again, it is coupled with ATP synthesis. Okay, and then the hydrogen ion ultimately is going to combine with the electron with NADP to produce reduced NADP. Okay, so here reduced NADP is produced at the end. Okay, reduced NADP is produced at the end. And one thing I want to mention here is you will see that this is slightly opposite of what happened in oxidation of in cellular respiration, because in cell respiration, first thing that happened is that reduced NAD will split to give you electron and H plus. Here, this is happening at the last step where electron and H plus combine to form reduced NADP. Okay, so but later we'll see more of this comparison later. Okay, and for cyclic, it is only for cyclic photophosphorylation, we see what happened. Okay, so here photosystem 2 is not involved. Okay, in a way you can just ignore it. Okay, so but what happened is that this electron, okay, so again same thing, sunlight coming in will excite the electron of the chlorophyll, so this electron will go through an electron transport chain, and of course as electron is transported by the ETC, the electron transport chain, energy is released, used to pump hydrogen ion. Ultimately, this hydrogen ion will be used for ATP synthesis. The only difference is that ultimately this electron then will come back to this PS1. Okay, so the electron that has been excited from PS1, the photosystem 1, will be accepted by photosystem 1. That's why it is called cyclic. Okay, but here the electron is not, you see, it's a one-way route. Okay, so electron from PS1, of course, you go to reduce NADP to give you reduce NADP, and the electron here will come from PS2, and the electron loss from PS2 will come from water. Okay, so it is not a cycle. You see, it's a one-way route. Okay, there's nothing cyclical in this, so that's why it is called non-cyclic photophosphorylation. Okay, so And then this is called cyclic photophosphorylation because PS2 is not involved, but only PS1. Okay, and also the other thing that's different here, because this electron ultimately is received by PS1 and not by H plus, and therefore there is no NADPH produced. There is no reduced NADP being produced. Okay, so that is the difference. Okay, so I think there should be a comparison. Okay, we'll come to see that comparison later. Okay, so here just another diagram showing you the whole process. Okay, so for the non-cyclic one, like we mentioned just now, okay, water will absorb some energy, okay, and then undergo photolysis, okay, from the sunlight, okay. That electron will be converted to will be accepted ultimately by photosystem 2. And for photosystem 2, when it absorbs sunlight, electron will be excited, and that electron will get excited and then transported across electron transport chain. And of course, some of that energy ultimately used to pump H plus and then form ATP. And ultimately, the electron will be received by photosystem 1. Okay, but of course, photosystem 1 would also undergo excitation where the electron will be excited. Okay, so maybe we start from, if we start from here, what happened is for the non-cyclic part, photosystem 1 absorb light energy, electron get excited, and then transport across electron transport chain. Okay, so here, part of it, you can kind of say there's ATP being produced as well, okay? And ultimately, that electron from photosystem 1 will be accepted by H plus combined with NADP to form reduced NADP. At the same time, this photosystem 2 absorb energy, electron get excited, transported across electron transport chain, and that electron ultimately received by photosystem 1, replacing what was lost from photosystem 1. Okay, and then the electron lost from photosystem 2 is replaced by electron from water. Okay, so that is a non-cyclic part. Okay, for the cyclical part, okay, so photosystem 1 is excited, okay, electron is transferred. It will go to electron transport chain, of course. Once it goes to electron transport chain, energy will be released ultimately to ATP production, and that electron is received by photosystem 1. So that's why it is cyclical. Okay, that's it. Okay, so this is just a diagram, okay, summarizing the same thing. Okay, you can just study it on your own. Okay, so I know this whole process looks a bit messy when you first look at it. Okay, so, and yeah, don't worry. Okay, so you just go back to it. Okay, you just have to read through it. Okay, so just get the logic right. But I think some of the logic is not unfamiliar to you because, like we mentioned, that logic involves electron going through transport chain, releasing energy to pump hydrogen ion, and the hydrogen ion will diffuse through the ATP synthase of the stop particle to form ATP. Okay, so again here, if you look at the comparison of cyclic and non-cyclic photophosphorylation, okay, so your non-cyclic photophosphorylation, as we see, involve both PS1 and PS2, but cyclic only PS1. Okay, and generally, of course, you would ask, what is the purpose? Why do we have two different mode of photophosphorylation? Okay, so if you notice just now that, okay, so in the case of cyclic photophosphorylation, no NADP is produced. Okay, so we'll talk about later. Okay, so NADP is actually normally for ultimately reduced NADP is for making glucose. Okay, so therefore, if the plant needs only more ATP, if it doesn't need the glucose, for example, okay, it doesn't have to carry out the glucose synthesis process, then it will carry out mainly non-cyclic photophosphorylation. Okay, so sorry, this is when there is no need for glucose synthesis, or maybe when there is not enough carbon dioxide. So when there is no carbon dioxide or not carbon dioxide is in short supply, then we make just ATP. But when there's carbon dioxide, we can make ATP and glucose. And to make glucose, you need reduced NADP. Okay, so that is the reason. Okay, so the reduced NADP helps make glucose eventually. Of course, formation of glucose require carbon dioxide as a raw material. So that's why whether this raw material is available would also dictate, okay, which process will be carried out. Okay, and ultimately the electrons from the photosystem will be used to reduce NADP, but here no, it's just returning back to PS1. So NADP is not reduced. okay? And so here's case, so of course with, and also you would realize that okay, in a non-cyclic process, there is no photolysis of water. No photolysis of water is involved in this cyclic process. That's why no oxygen is formed, only ATP. So this distinction you need to know. Okay, so photolysis of water, we already mentioned that. Okay, so splitting of water is a direct consequence of light causing chlorophyll molecule to lose electron. Okay, so because the reason is that the PS2 probably not depicted in a diagram. Okay, so PS2 also has is also combined with the water splitting enzyme. Okay, so when PS2 absorb energy, part of the energy will be used to catalyze. Okay, will be used to cause photolysis of water with the action of this water splitting enzyme. Okay, so oxygen is deemed as a waste product, as in something that's not required, will be released. Okay, and then hydrogen ion, as we learn eventually, will be combined with electron and with NADP to give NADP. Okay, but clearly that electron doesn't. This is not something that happens right after photolysis. Okay, what happens right after really is that that electron coming from photolysis will going through the whole cycle, ultimately then only combine with H plus with reduced NADP. Okay, it doesn't happen here. Okay, so photolysis, okay, photolysis of water will give you electron that go through this whole process, then at the end combine with H plus and NADP to form reduced NADP. Okay, so later we'll see how this reduced NADP is involved in this light independent reaction for the synthesis of glucose. Okay, so the synthesis of glucose is the light independent part. Okay, so yeah, photolysis of water can be demonstrated by Hugh reaction. Okay, so what is Hugh reaction? Okay, so basically it is a biochemical experiment. Okay, biochemical reaction that kind of mimics what happened in the chloroplast. Okay, so this Robert Hugh, he was a guy who discovered that oxygen is actually given off, okay, when you illuminate suspension of chloroplast. Okay, so which means, okay, of course, illuminate means shining light on it. Okay, so if you provide light energy, then of course chloroplast will photosynthesize and produce oxygen. Okay, so you want to check that what he did is that, okay, so because hold on. Okay, so long story short, what they do is that in this experiment, he want to show that, okay, there is what is it called? Hold on. I'll repeat what I say. Okay, so what happened, as we know, chloroplast release electron after receiving energy, right? Okay, so when you release electron, yourself is oxidized, you're actually exhibiting a reducing power. Okay, so for example, in chemistry, you learn like metal, they are strong reducing agent because they donate electron. Okay, so then, okay, so what we learn in this process is that ultimately there will be an electron acceptor. Okay, so in the case of Hugh reaction, okay, so basically he just check, okay, maybe if I put another, if I get an oxygen, electron acceptor, I can demonstrate this experiment, okay, or maybe prove a point that, okay, biochemically that is what happened. So if he replaced NADP with something else, okay, which can accept electron, which is an oxidized DCPIP. Okay, so DCPIP is blue in color, okay, in its oxidized form. So once when they sleeve extract chloroplast in light, electron is released. So this electron is going to reduce it. Once this DCPIP is reduced, it becomes colorless. Okay, so expect when performing this Hugh reaction experiment, you would expect to see a change in color from a blue DCPIP solution to a colorless DCPIP solution. Okay, so that is what happened there. Okay, so here just an experimental setup. Okay, so if you look at this particular, okay, so there are two different situations. Okay, so you do chloroplast in light and chloroplast in dark. Okay, so at first when you keep it in the dark, okay, if you look at this, no matter how long you do, so this is a colorimetric measurement, basically measure the intensity of blue color. So as it moves down, basically it is being decolorized. Okay, so if you keep the chloroplast in the dark, okay, without any, okay, in the dark, then the blue color intensity doesn't change. Okay, Of course, we know because there's no photosynthesis, electron is not excited from chlorophyll, so therefore electron is not released to reduce DCPIP. That's why color intensity doesn't change, okay? But once they're exposed to sunlight, okay, here this is exposed to sunlight or light, okay, this after this is exposed later. In any case, in both cases, okay, the presence of light, okay, causes formation of release of electron, which reduces your oxidized DCPIP and therefore turn it colorless. That's why over time you can see that, okay, you can see that firstly it actually drops in color intensity because DCPIP is accepting electrons being reduced over the process, okay? So here, why is there a downward trend? Okay, so this is okay. So your chloroplast absorb light and split water, generating hydrogen ion electron. So this electron reduce DCPIP, okay? So therefore, okay, so here, yeah. So, but this is not a very accurate sentence, which reduce DCPIP. Forming which is colorless. Okay, so that would be the explanation. Okay, so next, if you look at here, there's also a difference between the two curve. Okay, so why is that the case? Okay, if you look, okay, so of course you learn that in chemistry, you learn about rate, or even in bio as well. Okay, so we know the steeper the change, okay, the faster the rate. So clearly, here you can see that this rate is higher than this. Why is there a lower rate? Okay, so one possible explanation is that as you keep the chloroplast, okay, in the suspension, okay, but you need to know that after all, when you perform this experiment, you're isolating chloroplast from its natural environment. What can happen is that when you are put in an unnatural environment, of course, damage could be caused. Okay, so some of the chloroplast may be broken down, may burst, you know, for example, maybe the osmosis is not right or whatever. So there will be damage caused to the chloroplast. So therefore, for that reason, some of the chloroplasts are damaged, and that's why the rate of the photosynthesis is lower. Okay, so for example, when you place it in the dark, okay, but it has nothing to do with the darkness, simply because you're putting it there, okay, not doing anything, and in a non-natural environment, okay, for sure, some of them would be damaged, and therefore it's less thick. Okay. Okay, of course, and also here, it is also trying to explain this trend. Okay, so here it is steady. Why is it steady? Because in the dark, it doesn't reduce DCPIP. So that's why it doesn't release electrons. So DCPIP not reduced. So therefore, color intensity remain largely unchanged. Okay, so that is a reason why. For this one, we'll skip this one. Yeah, because this one I need to find a YouTube video for it. I'm not sure which one it is. I think there was one. I'm going to find you this video later. Okay. Next. Okay, now let's look at some of the comparison between photophosphorylation and oxidative phosphorylation. Okay, so what you see is that, okay, so of course, location is different. Okay, so photophosphorylation happen in chloroplast, and then phosphorylation is mitochondria. If we talk about a very specific location, it is thylakoid membrane in the case of photophosphorylation, and inner membrane of mitochondria for the case of oxidative phosphorylation. Okay, so involvement of light energy, of course, okay, for oxidative phosphorylation, no light energy is required, but then it is required for photophosphorylation. But we need to emphasize, okay, so okay, so of course, light energy is required. So it is mean, okay, for splitting water, or in fact, not just for splitting water, okay, also for electron excitation. Okay, so this electron excitation, okay, that's from chlorophyll. Okay, source of energy, of course, from light. This is from the oxidation of glucose. Okay, And then electron donor, okay, so for the non-cyclic, okay, it is water. Ultimately, electron come from water. Okay, but for cyclic, it is just PS1 returning electron back to itself. Okay, and then in the case of oxidative phosphorylation, ultimately the electron, okay, in the case of respiration, we remember the case, so it's a reduced NAD that's going to contribute H, which split into electrons. So it is the electron donor in the case of oxidative phosphorylation. Okay, and then the electron acceptor here is for electron acceptor in the case of photophosphorylation is NADP for non-cyclic, okay, PS1 for cyclic. Okay, for oxidative phosphorylation, as we know, the final electron acceptor is oxygen. Okay, this is about the direction of pumping. Okay, so here protons are pumped inwards. Okay, so it's a diagram that I showed you just now. Okay, so in this case, where is it? Okay, so your proton is pumped inwards. Okay, proton is pumped inwards into the mitochondria. Okay, sorry. In the case of mitochondria, it is pumped outwards. Okay, so from the matrix out into the intermembrane space. For the case of chloroplast, it is pumped inwards from stroma into the into thylakoid space. Okay, so those are the comparison. Okay, so next we'll look at light independent reaction. Okay, so light independent reaction, later we are going to see, it's kind of, again, there is some similarity to your cellular respiration. Okay, in cellular respiration we have Krebs cycle. Okay, and light independent reaction we have a Kelvin cycle. As you'll probably expect, okay, this Kelvin cycle also involve conversion of multiple organic compound from one form to the other, and eventually it forms a cycle. Okay, of course, the whole process eventually is going to lead to production of a carbonic, so it's a carbon-based organic compound, which is glucose. Okay, so here, okay, so the place where it occurs is in stroma, just like Kelvin cycle in matrix. Okay, Krebs cycle happened in matrix of mitochondria. Kelvin cycle happens in the stroma of the chloroplast. Okay, and energy, whatever that is generated from the light dependent reaction will be used. Okay, so actually kind of we already implied it just now. Okay, so just now, at the case of non-cyclic photophosphorylation, just now we have learned that, okay, you produce ATP and reduce NADP. Just now we also say this reduced NADP is required for making glucose. Okay, so yeah, that's how it is involved. Okay, so it takes place in stroma, and because this has been performed, okay, in the presence of light, okay, whatever that happens next doesn't require light. Okay, so actually, for your information, the process of photosynthesis, okay, can continue at night. It's just that at night there is no photolysis, there is no no oxygen being produced. Okay, but it doesn't mean glucose is not synthesized. Okay, so if by photosynthesis we include the process of glucose synthesis, okay, at least half of the process can happen at night. Okay, so that's why it does not require light directly, and therefore it's also called the light independent or dark reaction. Okay, so there are three main stages, namely fixation of carbon dioxide. Okay, so basically it means you fix carbon dioxide with something, with an organic compound that is present originally in the plant. Okay, so we'll look at more detail later. Okay, so next is so ultimately what you get is a 3-carbon glucose phosphate. Okay, sorry, it's I don't remember the name. Okay, we'll come to it. Okay, so it's a 3-carbon compound, and this 3-carbon compound will be reduced eventually to produce a triose phosphate, and then okay, so there will also be something that is regenerated. Okay, there's also something that's regenerated. Okay, by the way, this triose phosphate, this is also triose means 3-carbon. So and then 2-triose phosphate will give you glucose. Okay, it will give you glucose. Okay, and of course, it's a cycle. Something need to be regenerated. So it is this RABP. We come to talk about it in a minute. Okay, but here a few things I want you to see. Again, if you remember, for the case of Krebs cycle, you go from glucose. Actually, you get 2 times 3-carbon compound like triose phosphate. and then it go through the Krebs cycle, and it is an oxidation process. Okay, and then you will learn that, okay, here the okay, so when we say oxidation, it means hydrogen is released, so you get NAD to become, I'll just do NADH. Q2 means reduce NADH. Okay, so that is what happened during glucose oxidation. Okay, but for the Krebs cycle, it's a reverse. Okay, NADPH will be oxidized to become NADP. The hydrogen is used to reduce. Okay, so the Calvin cycle is essentially a reduction process. Okay, it is a reduction process. Okay, so the compound is reduced multiple times, and that reduction is due to acceptance of hydrogen from NADPH. Okay, so of course ultimately you get two triosphosphate, okay, something 2,3 carbon compound, which will combine to form a glucose. You can see it's kind of the reverse of the whole thing. Okay, but I'll come to look at this in more detail later again. Okay, so yeah. We'll continue this in the next video.